Modern medicine can seem like magic. You can buy a pill over the counter that stops you from feeling any pain, treat cancer with tiny localized bits of radiation, and even reduce your risk of heart disease by treating the liver. But how? I'm glad you asked, because we'll learn all about it in this paper. In vivo CRISPR base editing of PCSK9 durably lowers cholesterol in primates. They made a unique type of nanoparticle that removes harmful proteins in the liver that cause plaque buildup in your arteries. Plaque that would otherwise put people at a very high risk of cardiovascular disease. And here at Brain Rot University, we want the significance of this paper to be painfully clear, which is why we get a brief explanation from me and from our good friends at pdftobrainrot.org. They transform regular, dense scientific papers into a form factor that everyone can understand. A Subway Surfers clip with Sigma, Skibbity, and Goon used an uncomfortable amount of times. Once it makes that, we're gonna grill the hell out of this AI's explanation to see if it got any of the science right, and if their explanation was better than mine. So without further ado, class is in session. Like with all papers, we need to go over some of the important background information as well as the big problems that these authors tried to solve. But don't worry, my cute little nerds, I'll keep it as non-technical as possible and really only expand on the important bits. First things first is we need to talk about cholesterol, specifically low density lipoproteins, or LDL. This is what many consider as bad cholesterol because it commonly sticks to the inside of the blood vessels and causes plaque buildup. And too much plaque buildup can block arteries, which eventually leads to heart attack or stroke. In fact, plaque buildup, also known as atherosclerosis, is the leading cause of death worldwide, being linked to an estimated 30% of all deaths. I hope it's clear that reducing your LDL is of utmost importance for reducing your risk of cardiovascular disease. Luckily for us, our body naturally tries to minimize the amount of LDL in our body through a number of different ways. The main ones outlined in this paper are through something called LDL Receptor, or LDLR. It's a receptor that's found on cells all throughout our body, with a whole lot extra being in the liver. And it works something like this. LDLR binds to little LDL particles that are circulating in our blood. This brings them to the liver, which can then degrade the LDL in order to prevent them from doing any harm. Alright, so problem solved. Our bodies have a way of dealing with the LDL, and they can clear it when it gets too high, so no one will ever get heart disease ever again, right? Right. Now it's time to introduce the antagonist of our story. The dark, mischievous, mysterious proprotein convertase subtilis kexin type 9. We're gonna call him PCSK9. PCSK9 is a bad guy because it destroys LDLR. Remember, that's the good one. And here's how it works. When there's a lot of PCSK9 in and around the cells, it signals those cells to destroy LDLR. This usually happens when there are too many receptors. And this is fine if there is balance and everything is maintained in homeostasis. But if PCSK9 is too active, too many LDLR will be destroyed, which can let LDL wreak havoc on our arteries. So in summary, as PCSK9 increases, LDLR decreases, and thus LDL and eventually cardiovascular disease also increases. So what's anyone doing about this? Luckily, there are a few different treatments that are available, such as the ones that are shown here, that help to reduce the LDL cholesterol. Some, but not all, act directly on PCSK9. And though they might be good, they're not great. They often need many repeated doses in order to be effective, which if any one of you have ever had a grandparent or had to take antibiotics for an extended period of time, you know that humans aren't always great at sticking to their medication schedule. So instead of a treatment that needs temporary yet repeated doses, these authors tried going to the source. These authors made a once and done solution by editing the genes that code for this PCSK9 protein. And they did so by using two technologies that are very common in today's lexicon: CRISPR Cas9 and lipid nanoparticles, also known as LNPs. Now, in case you've been living under a non-scientific rock, CRISPR Cas9 is a very precise and relatively easy way to edit genes. Scientists add these things called guides to locate exact genes they want to edit. The Cas9, which is an enzyme, simply then cuts these genes. Very recently, this technology was famously used to de-extinct certain prehistoric animals, such as woolly mammoths and direwolves. They did this by CRISPR Cas9ing these once living genes into currently living animals. Lipid nanoparticles, or LNPs, are just what they sound like. They're nanoparticles made of different combinations of lipids. And for the purpose of this paper and other similar papers in this field, they're really used to package and deliver the different components of the CRISPR Cas9 system. Now, in my opinion, this is where this paper gets very interesting, but in a very pragmatic way. LNPs love going to the liver. Like, over 90% of them get there within an hour. We don't really need to go over the specifics today, but if you're a little nano nerd and you want to learn more, subscribe to the channel, come follow me on Instagram. We talk about this stuff all the time. What you do need to know is that these authors made an LNP with the CRISPR Cas9 components in it. This CRISPR Cas9 system was designed to specifically knock out or reduce the amount of PCSK9 that the liver produced. By my calculations, the LNPs, they like going to the liver, and PCSK9, which is what the LNPs are trying to knock out, is also made a lot in the liver, which is where the LNPs like to go. Huh? Say bye-bye, PCSK9. Sure, this sounds nice in theory, but how effective was it really? After injecting these LNPs into monkeys, PCSK9 was reduced by up to 81%, with that stinky LDL being reduced by around 65%. The most impressive parts? These results held up for up to 8 months, with no side effects noticed in any of the monkeys tested, all with only a single injection. Now, if you remember what we said before, current treatments need repeated doses, typically 1 pill every single day, sometimes multiple if you're eating a lot of red meat. The fact that this showed you only need 1 dose to last many months is super impressive and a really big improvement over current treatments. Now, I hope that was clear, but in case it wasn't, let me try again in simpler terms. LDL blocks your arteries and can cause heart disease. LDL receptors, or LDLR, blocks LDL from causing harm. But PCSK9 blocks you from keeping you healthy. All this happens in the liver. Thanks to CRISPR gene editing that's packaged in an LNP, which loves to get to the liver, these authors could stop PCSK9 and LDL long term after just a single dose. The end. We're going to upload this paper into the site. It's going to make a little brain rot video. Then we'll watch it a second time, go line by line, and try and figure out what the brain rot was trying to say about this paper. Critique it along the way, see if it was even accurate. If we're lucky, this video will be so bad that we'll have to upload it again and see if it can do it better a second time. You know, the last time I made one of these videos, I ran out of credits on this site and had to buy more. So it's good that we're getting our money's worth. Remember, this paper's called In Vivo CRISPR Base Editing of PCSK9 Durably Lowers Cholesterol in Primates, Gene Editing for Cholesterol Reduction in Primates. You know what? This site always does a great job with the titles. I feel like they always nail it. And now for real, class is in session.
Gene editing for cholesterol reduction in primates. Speedy bop. Let us dive into the dank world of gene editing and cholesterol. Scientists, low-key gooning with CRISPR magic, have delivered lipid nanoparticles to primate livers, turning the tide against PCSK9 like a Sigma boss. With a single infusion, they slashed cholesterol levels by around 60%, keeping it chill for 8 months. Imagine a toilet that flushes out bad cholesterol. That is the edge of science right here. This is not just a ship sailing in circles. It is a one-and-done strategy to tackle cardiovascular woes, proving that gene editing tech can be a total game changer in health.
I don't want to say too much. I prefer not to speak. Because if I speak, I'm in trouble. This might be the best first round that we've ever seen in the series. As you can tell by my reaction, there was some stuff I was not impressed with, but that was really good so far. Let's try it again. Let's see.
Gene editing for cholesterol reduction in primates. Let us dive into the dank world of gene editing and cholesterol. The tide against PCSK9 like a Sigma boss.
A Sigma boss, because that's exactly what LNPs are.
With a single infusion, they slashed cholesterol levels by around 60%.
Okay, so that is by far the best way they've introduced data into any of the videos that I've seen. Usually they kind of throw it at the end of the brain rot video, and it doesn't always do a good job of like linking the different concepts together. But so far, I feel like this has been very clear in terms of, hey, what are they trying to do? Like, what are the LNPs being made for? Where are the LNPs going? And what is their effect? I think the only thing that they haven't really linked is like, what is PCSK9? I think up until this point, all they've said is PCSK9 is there. And what was it? Something about like, it slashes PCSK9 like a Sigma boss or something like that, which again is true. But if you're just scrolling through TikTok, no one knows what that is. Like, no one's going to understand what is PCSK9. Like, and shockingly, that's probably because the brain rot doesn't actually understand what's going on. It just recognizes that PCSK9 is in the abstract, and that's what this paper's trying to do. All that being said, the 60% reduction, really good. Really, really impressed that they showed that.
Keeping it chill for 8 months.
Pretty chill.
Imagine a toilet that flushes out bad cholesterol. That is the of science right here. Okay.
These videos are always trying to loop in toilets, and I get it, all right? Skippity toilet's a thing. As soon as I hear toilet, I'm out. And then you could tell by my first reaction, I was out.
Maybe so, sir. But not today.
It wasn't just a stupid Skibbity reference. They were actually trying to make an analogy for why These LNPs are like a toilet. What do they say? What were the exact words?
That is the edge of... Imagine a toilet that flushes out bad cholesterol.
Flushes out bad cholesterol.
That is the edge of...
That's true. That's what the LNPs are trying to do. They're trying to get rid of that bad cholesterol. They're trying to get rid of the upstream effects, which then leads to those downstream bad cholesterol. So it actually is right. Honestly, super impressive. Okay, the other thing is they're trying to be funny with edge, because edge is fucking hilarious. We all can agree on that, but not when it's used properly. You know what I mean? Not when it's used for like what? Normal colloquialism of edge of science. Boring. I want to see them talking about primate livers edging. All right?
This is not just a ship sailing in circles. It is a one-and-done strategy to tackle cardiovascular woes, proving that gene editing tech can be a total game changer in health.
It's like they've been watching my videos. I'm convinced that they've been watching my videos. Or that's just how LLMs work. It's like the more you upload stuff, the more it learns how to better communicate it. Anyways, they always try and talk about ships. All right, in every video they talk about ships. And once again, this is the first time where it actually makes sense. For this one, just like with the toilet, it has a good ship analogy. Instead of it being a ship that's going in circles around and around, just like what current treatments need to do, which is needing many, many rotations, many, many sails in order to complete its trip, it just needs the one. This ship, this LNP ship, just needs the one dose to be effective for a long time. Which is, what do they call it?
Editing tech can be a total game
Proving
changer
that gene editing
in health.
tech can be a total game changer in health. Yes, that is exactly what it is. We have a brain rot university first. We only needed to do one video before it wasn't trash. Wow. Good job, everyone. You, yes. All right. Good job, team. All right, yes. Yeah. Now, I think there are a few reasons why. One is it's an LLM, so it probably understands, hey, the more that this stupid account keeps feeding me nanoparticle-related stuff, the more I can understand what's actually going on. Two is that this paper was a lot simpler than what we've done in previous Brain Rot University lectures. Not to say that this was super straightforward, like unless you're someone like myself who's been studying nanoparticles for a long time, a lot of this information is probably new for most people. However, it's a lot more straightforward than what we've seen before. This paper used a lot of scientific words that are now common in today's lexicon. For example, lipid nanoparticles or LNPs, they've been super popular since the pandemic. CRISPR Cas9, they've kind of been science's darling for like at least a decade now. So it probably makes sense that the AI had more to draw on with these simple concepts. Pretty straightforward. In the past, not like my research was super groundbreaking and unbelievable, but it's definitely more complicated. There was that layer of complication that really only nanoparticle nerds would get. That was kind of where their value was in their level of complexity. So yeah, other than them not really linking PCSK9 all that well, this did a great job. Like I think it would have kept my attention. I probably would have kept going with this if I saw it on TikTok. It definitely would have given me enough to be like, hey, is that real? You know, I'm biased, but I don't think it was better than my explanation. But that's not for me to decide. That's for you to decide. So you let me know down in the comments who did a better job. Dr. Matty C, PhD, Professor Brainrot AI Slop. You can also let me know down below other papers you'd like me to cover. I like to stick within the biomedical engineering sphere, but hey, I'm down to try anything. If you learned something today, give this video a like. If you learned a lot today, think about subscribing. Also, come join me over on Instagram. That's where we have the biggest and best community in the whole nanoparticle world. I'm also going to talk about totally non-scientific things, such as why do I go to prison for vacation? But that's all for today. Thank you for stopping by, and see you later, nerds.